Thanks very much, Nick, for that fantastic introduction. It's a real pleasure to be here in these fantastic setting of the Royal Institution. So I'm going to talk today about the neural and neurochemical correlates of OCD and where that may lead us in terms of new treatments, which I think are very important. So OCD obviously is expressed in a number of ways, but we theorize that there are some common elements in OCD in terms of actual mechanisms in the brain. And for example, we know that OCD patients have emotional vulnerability. Obviously, anxiety and response to uncertainty is a very key issue. Equally, we think there are some cognitive factors which are important. For example, loss of cognitive control, which is evident in the sense of the loss of control over symptoms, for example, compulsive behaviours and intrusive thoughts. And we also think that this applies in particular to a balance in the brain between two systems, a system which controls our conscious voluntary goals and plans, and a more automatic system which controls our involuntary behaviours, our automatic behaviours, which are very important to us and make us very efficient. But in a sense, in OCD, we think there may be a bias towards that system, which results in this repetitious behaviours. And these factors combine, interact to produce OCD in its various expressions. Now, there's been over 30 or 40 years' work of brain imaging of OCD in various brain scanning modalities. And there is now a consensus emerging about what happens in the brain in OCD. Either this is happening before the symptoms start and then cause the symptoms, or certainly it's present during the expression of the symptoms. And this is a diagram which Mathilde Vagui, who's now at the Birkbeck College in London, University of London, and I constructed a few years ago, which illustrates the concept which we've come to, that there's an imbalance in what we call frontal lobe systems. So the frontal lobes, as you realise, are behind the eyes here. And they comprise a number of structures, actually, which interact like a committee to determine our behaviours. And they control other areas, lower areas, if you like, in the brain, which express these behaviours. And just to simplify things very dramatically, I'm going to argue that the imbalance is that there tends to be an overactivity of what we call medial areas of the frontal lobes, immediately behind the eyes, and an underactivity of more lateral regions on the side here. And this interaction between these two areas governs the balance between goal-directed and habitual behaviours. And as you see, these systems control these so-called subcortical areas involved in disorders like Parkinson's disease and Huntington's disease, which express behaviour and are called the basal ganglia or the striatum, the caudate nucleus and the putamen. So just focusing in particular on this medial system, we did a study a number of years ago using aversive conditioning. So some very brave OCD patients volunteered to perform in a behaviour where they had to avoid mild electric shocks. And while they were doing this, we scanned their brain and measured the way in which essentially blood flow represents a proxy of neural activity in different areas to find out which areas were active in these avoidance behaviours. So this is a rather artificial lab situation, but it's the best you can kind of do in this research. And what we found indeed is that this area, the behind the eyes, was extremely active during this active avoidance behaviour in OCD, much more active than in healthy, if you like, volunteers, control subjects. And then we did some rather clever correlational analysis and identified a network which was hyperactive. And you can see the network here, which includes this structure here, the so-called dorsal anterior cingulate cortex. Sorry for that technical, there's no other way of describing it. But it's kind of in the midline here, just behind the frontal lobes, and quite close to an area called the supplementary motor area, which controls these structures expressing behaviour. And as you can see, in the OCD patients, these areas are much more active than in controls. Now, why is this interesting? Well, it's interesting because a lot of other research has suggested that this area is particularly overactive in OCD from a whole variety of different lines of evidence. It's been associated in experimental animals with functions like checking and monitoring behaviours and really controlling the difference between goal-directed and habitual behaviours. So there's a basic science underlying this. But in OCD, we see several signs of this. So one sign is the so-called error-related negativity. So this is using electroencephalography to measure evoked potentials from the scalp of the brain. So this is not invasive, these are scalp electrodes. And Anna Maria here is an expert in this area. You may have met her in Naomi's clinic. And basically, when errors occur in a task, this error-related activity gets bigger. But it gets really big in OCD patients. It's almost like a marker. And it's thought to represent activity in this anterior cingulate area. And there's other evidence that this region is very responsive to uncertainty and doubt. Interestingly, it's a target for neurosurgery in OCD. Now, it may surprise you to learn that really the only case where neurosurgery has ever been found to be effective in mental health disorders is in OCD. And what is the target? It's the dorsal anterior cingulate. So bizarrely, it's still performed quite regularly in very, very severe patients. They receive small lesions of this area, which obviously dampens its activity. And it can be very effective. Brain imaging data also suggests that checking, compulsive checking, is very much linked to overactivity in this area. So you see, I'm building up this case that this area is just too active, the balance has gone wrong. What does this mean in terms of the neurochemistry or the physiology of the brain? Well, in your cortex, in the cerebral cortex, in your frontal lobes, the balance of activity is controlled by two neurochemical messengers in the brain. One is called glutamate, which you've probably all heard about, glutamate, which is excitatory, so it's an on switch for a cortical network. And the other is called GABA, which is short for gamma-amino-butyric acid, sorry about that. And that's inhibitory. So glutamate GABA, the ratio is a balance, which hypothetically is towards glutamate in OCD. But no one's really ever convincingly demonstrated that. Now, why is this interesting? Well, you know, there is some genetics of OCD which has suggested that some genes which are involved in regulating glutamate go wrong. And this has been tested in mice, for example, where the gene has been knocked out, so it doesn't work. And some of these mice groom a lot, which is a kind of very simple primitive behaviour, which, who knows, might be related in some sense to OCD. So we performed a big study a couple of years ago, funded by the Wellcome Trust. This is the team we had, so we had a fantastic team. Susanna Maria here, Paula Banker, who's a postdoctoral fellow, Marjan Berea, who took the lead. She's doing a doctorate in clinical psychology, and I think she's still interested in OCD. And Murray Healy's just finishing her PhD in Cambridge, and she's following up some of this work, which we performed in a 7 Tesla magnet. So there are very few 7 Tesla magnets actually in the world. I think there's only a couple in the UK. They're very, very strong magnets. And the reason we use a 7 Tesla magnet is you need it for this kind of research, where you're actually going to use imaging to measure neurochemicals in the brain. So how does it do that? Well, basically chemicals resonate when a magnetic force is applied to them, and you can measure the frequency of that resonance, which is specific to each chemical compound. So you can actually quantify how much neurochemical there is in the brain from this magnetic spectroscopy. Incredible technique. And basically we tested about 30 healthy volunteers and again 30 very brave OCD patients. So it's quite a small scanner this. You have to really scrunch up and you have to be very still and not move your head. And we also had to screen very carefully the OCD patients who were able to perform in the task, because not all of our volunteers could be accepted for various reasons. So let me show you some data from this study. So this is the kind of records you get. You can see these spectra of neurochemicals. So the height of the reading demonstrates how much there is in the brain, basically. So you can convert that directly to actual measures of the amount of chemical. And the amazing thing about the 7 Tesla magnet is that you can discriminate glutamate from some of its metabolites, which you can't do very easily with lower magnet strengths. That's why this is so unique. So these are very accurate measures of glutamate. And also GABA. And you can see some of them separated here in this record. Now, where did we measure? We measured in 3 areas only, 3 voxels, as it's called. And one of the main voxels was in the supplementary motor area, which is a big output of the anterior cingulate. The anterior cingulate projects to it and sends its messages there. And the supplementary motor area is the kind of head nerve center of the habit system in the brain. It controls our habits. And what did we find? Well, these are correlational maps at the bottom here, and what you see on the x-axis is the concentration of glutamate, the actual chemical concentration of the messenger. And on the y-axis, you can see various measures. Now let's first of all deal with the Y-BOCS scale, which you're all familiar with. Is a measure of severity of OCD symptoms. And these points are individual patients with OCD, the 30 or so, with their various Y-BOCS scores. And you can see that there's a relationship. Whenever you see a gradient like this, there's a positive relationship of the level of glutamate with the Y-BOCS severity scale for compulsive behaviour. That's an amazing result. This is statistically highly significant. It's an amazing result. A correlation between a neurochemical in the brain and your symptoms. I mean, this is amazing. Okay? Highly significant, 0.01. So the chances of getting that correlation by chance are 1 in 100. And we normally take 1 in 20 to be significant. Okay? Additionally, we measured OCI. OCI is the Obsessive Compulsive Inventory, which is a questionnaire that we give to OCD patients, but also healthy volunteers take this as well. And you know, we've all got a bit of OCD, as they say. Certainly we scientists do. We score on the OCI scale. And as you can see, the OCI scale gives us the same answer for patients in blue here. The same correlation. And look at this. Even in healthy volunteers who score much lower on OCI, there is a relationship between glutamate in this region and their OCI score, their degree of mild compulsivity. It's an extraordinary result. And this is one of the reasons it was published in Nature Communications, which is a very, very prestigious journal. Okay, so that's quite interesting. But what about the anterior cingulate, you're asking me? Well, we didn't see relationship directly with symptoms there, but what we did see dramatically is, first of all, a highly significant increase in glutamate levels there, and a highly significant reduction in GABA levels. So just think about this. Glutamate goes up, that's the excitation. GABA goes down, so the ratio is enormously increased in favour of glutamate. So this is the reason the anterior cingulate is more active. And guess what? Just as we found actually also for the SMA, We also gave a task, a behavioural task in the scanner, which measures the balance between goal-directed behaviour and habits. I'm not going to go into details, but I will if you're interested. Okay? So this is a computer-driven task here, which subjects do before they go into the scanner. And look at this. This is a measure of the glutamate-GABA ratio here against the bias to habit. This is called the habit responsivity index. Highly significant correlation in OCD patients. So the more glutamate in the anterior cingulate in OCD, the more habitual you are. So that presumably provides a cue to the supplementary motor area to drive this habitual automatic behaviour. So this is just in the artificial environment of a lab, of a scanner, but it does show this tendency, which is of course not present in healthy volunteers. If anything, it's the opposite, which is interesting. Okay, so we found this, and there is controversy in the field in science. There's always somebody who's determined to prove you wrong. So we have to look around for our supporters. You know, what is there in the literature which backs us up? Well, first of all, this is our study at the top. You know, a while ago, people who had lumbar punctures, I guess, with OCD, did find that glutamate levels were increased in OCD when they assayed them. So that's an old study, but it's consistent with our evidence. And there's been a couple of weaker magnet studies which kind of have backed us up. So there's a study in the States showing that there are increases in glutamate in the anterior cingulate in children with OCD even, which is intriguing because it provides possibly a trait-related factor. And also there's been some evidence from Columbia, New York, with reductions in GABA, which is the other result, in adults with OCD. Again, a weaker 3 Tesla study. But we're the first study that's shown both effects, up and down, up glutamate, down GABA in the anterior cingulate. So what does all this mean? And how does it fit in with OCD? Well, you know, the SSRI treatment, we don't really know how SSRI treatment works. We're still working this out, actually. You know, it works on a messenger called serotonin. But, you know, what does that do, actually? Well, one of the effects of SSRI treatment is to normalise levels of glutamate in the striatum. Okay? And maybe psilocybin works like that as well, possibly. And, you know, there is some interesting result with MRS that effective behaviour therapy actually has an effect on the anterior cingulate glutamate levels. There is a study showing that OCD patients with effective cognitive therapy show a normalisation of glutamate in the anterior cingulate. So that's amazing, but also encouraging, right? Okay. What about other treatments? Well, I've mentioned SSRIs, but there's a whole range of other drugs which have been tried in OCD to some limited extent in phase 2 trials. Memantine, riluzole, N-acetylcysteine, ketamine, lamotrigine, topiramate, glycine, maybe TDCS. Well, all of these have some reduction effects on glutamate. So this is kind of consistent because most of these phase 2 trials show some small effect, which is not quite significant or, you know, needs further work, and that's why they've never been adopted in a major way. There are also some very creative animal models of OCD focusing on glutamate, as I mentioned earlier. So, for example, the SAPAP3 knockout mouse has an elevated expression of glutamate receptors, and optogenetic stimulation actually can reduce compulsive grooming in this mouse. So that's interesting as a primitive animal model. I'm never sure what it's an animal model of because, you know, when the mouse is grooming, does it really think it's worried about germs? I don't think so, actually. But it may be a model, for example, of trichotillomania, which is quite a common human disorder. Okay, so we've got a strategy. And our strategy is to reduce glutamate more effectively than has been done previously. Is there a way of doing this pharmacologically? Yes. It's called the mGluR2 receptor. Oh, very complicated. So I'm going to really simplify this. These are two nerve cells, okay, two neurons, and the messenger goes from one to the other across this synapse or gap, if you like. And the receptors on this postsynaptic cell, which which receive the message here, these are glutamate receptors postsynaptically. But intriguingly, there are also receptors on the presynaptic cell which regulate the release of glutamate. So this is the way the brain, typical brain, regulates itself. So when this neuron is pulsing away, trying to be active, in order to prevent it getting too excited, there's a presynaptic receptor which glutamate binds to, which says, calm down, don't release glutamate. So when you bind to that receptor, when glutamate binds to that receptor, it calms the system down and it releases less glutamate. Okay? So let's give a drug which does that. And there are highly selective drugs, they're called positive allosteric modulators, which are really nice drugs. They're not rigid drugs, they flexibly help the receptor respond to glutamate and reduce glutamate release in the terminal regions. Okay, get it? Hopefully. Now, these drugs actually have been trialled in schizophrenia by Lilly and by AstraZeneca, for example. And you know, these big pharma, they always do the wrong trial. They tested chronic patients with schizophrenia, and the drugs didn't work. Okay. Well, there's very good reasons for that. One of the reasons is that in schizophrenia, we think that glutamate goes up initially and then goes down. So it would only work to begin with, which could be very important. And when Lilly did their retrospective analysis of the data, they showed that the drug did work in first episode patients. And they should have stuck with it, actually. And they've never done a trial since. They should come back to it. But anyway, the good news about these trials, perhaps the only good news, is that the drug did get into the brain, which is one thing. And secondly, it had an okay safety profile. The adverse effects weren't very much. So that's a good start for any drug, because if safety is good, at least you can try it out in other conditions. So it's not been tried in OCD, and we're trying to change that. But you have to do a lot of work with such a drug, and obviously one of the things you need to do is to have some kind of animal model where you can try the drug and try the various parameters of treatment and just check out the safety aspects and so forth and so on. And so we've been doing that in collaboration with Amy Milton here, using a novel animal model of OCD, which Amy is going to describe in great detail the rationale, and you'll have to quiz her about the rationale. But okay, let me try and give you a quick rationale. Check-in is one of the fundamental disorders in OCD, one of the fundamental symptoms. That's been shown actually formally in paper in psychological medicine. And it makes a lot of sense in terms of the anterior cingulate. Actually, we've measured check-in in human studies. Sharon Moraine, who's at Anglia Ruskin University now, did a study where basically what happens is you're trying to earn points for rewards, responding on two buttons or two options, and you can't predict which one is going to be the correct option. However, you can get information about this by responding on a third button, okay? So how much do you check your information? And she actually found in this study that OCD patients did check more, which is encouraging in this very artificial situation. But anyway, you can do exactly the same test in a rat. Amazing. You don't give them points or money, you give them food, okay? So rat responds for delicious sucrose pellets, perhaps, on a good day, between two levers here. They can't predict which one's going to provide the reward, but there is another option at the back of the chamber which they can check to give them a clue as to which one is next going to be correct. A light goes on above the to-be rewarded lever, okay? So the rats are pretty bright, you know, especially Cambridge rats. These are first-class honours rats, the hooded rat. They're very delightful creatures. But some of them do check a lot. They check more than what's good for them. They lose rewards because they get hung up on this checking behaviour. In a particular strain of rat, which Amy will describe. So we've tested both of these drugs, AZ, AstraZeneca, and AZ-08529, that's the way they call these drugs, in this procedure. So let me describe these data. So this is the check-in lever presses here. So these are the number of times the rat presses this lever, and these are the excessive pressing where they're checking much more than they need to. This is what we call a dose response curve. And these three histograms, the white histogram and the one after one, the first one and the third one, they're the baseline. So this is before and after the drug, okay? This is placebo, 0 milligrams per kilogram. And here you see 3 milligrams per kilogram and 10. And you can see this dramatic reduction of check-in behaviour. Fantastic. It works for excessive check-in as well. It's a really big effect, huge effect. And by the way, it's achieved with doses 20 times lower than found to be effective in the AstraZeneca models of schizophrenia they used, because these drug companies never really do things right. And you can see this work is in submission for publication. Now, the really interesting thing, from my point of view about this, is that the animal's behaviour in getting rewards wasn't much affected by these doses, because you might well say, oh, well, if the drug sedates the rat, perhaps the rat's lying on its back and can't check anymore. But this is not the case. They continue to try at the two levers and actually don't lose many rewards, except maybe at the highest dose here. So that normal performance carries on. And this is, rather selective anti-checking effect. Well, more recently, we've compared it with the Lilly drug. And the Lilly drug's also effective here, as you see. We just highlight these effects at the high dose. You can see, again, highly significant reductions in check-in. And also for excessive check-in. So we think this is a very promising approach, and we are trying to raise funds for a phase 2 trial of this compound, or one of these compounds. Okay, so my conclusions. Present results are consistent with this imbalance. The hyperactive circuit is consistent with altered excitatory inhibitory balance. The balance is related to changes in the excitatory messenger glutamate, can be adjusted by pharmacological agents, the mGlu2 agonists that reduce this release of glutamate. Existing pharmacological treatments may do this already to a limited extent. And in theory, other non-invasive neuromodulatory treatments may also work to reduce glutamate. I mentioned cognitive therapy and TDCS, but maybe transcranial magnetic stimulation will also do this if applied in the right way. So thank you for your attention.